Hello everybody, my name is Jack Rust, and this is our presentation on sustainability and the things that we've learned in our class.
I'm Isaiah, and I'm talking about healthy soil. Healthy soil is represented by multiple different organisms such as bacteria and protists that help contribute to the function of soil. Without the diversity of good and bad organisms, soil would fail and would cause widespread famine across the area affected. In order to sustain soil, we must not add fertilizers that kill off organisms, as it doesn't just get rid of the bad microbes, it also gets rid of the necessary ones. For our results, our whole class got together and got 6 different site locations, where each location will be shown on the next slide. For our data, we used plates to calculate how many unique organisms were found at each different location site. The photos show different sizes of fungi and bacteria that grew over a 5-day time span. The graph also shows the average amount of organisms at each site. Contrary to my original hypothesis, which was lawns and agricultural areas would have less biodiversity compared to forested areas, the opposite happened. There was actually more biodiversity in the agricultural areas. However, just because there is more diversity in a general area does not mean that it could show up in a different area. Microbial organisms can be good or bad for an ecosystem, which can be problematic for a certain area. In conclusion, our group found that results were not expected or similar to the hypothesis in any way. This map shows where each soil location was taken in the general vicinity. Martin Farm is located beside Parker Side Road. Does not make an appearance on Google Maps, but the general location is shown via satellite imagery.
I'm Maya, and I did research on silica in water. So first thing about healthy water is that it's not going to kill you if you drink it. That's the goal, is to not die. And unhealthy water, it will harm or kill you if you do drink it. But silica in water, I couldn't find anything that say silica is necessarily bad for you. It's just not recommended that you eat silica. But in some cases, you can be allergic to silica, which can include burning of the throat, swelling of the face, and other allergic reactions. And there are no federal regulations regarding silica in water, but a lot of states and local governments do have regulations regarding levels of silica in water. Silica can also be harmful to machines like dishwashers and washing machines because of all the buildup. And here is a map of where we got our water sample.
So a major question is, what even is sustainability? Sustainability is defined as meeting the needs of our current generation while also still maintaining and saving the environment around us for the next generation. This includes things like soil and water, which are very important for us, because how else do you get your crops to grow if you don't have any soil or healthy water? Now, how does this all relate to sustainability? By learning about how to be able to test your own soil and your own water, you can help make yourself a lot more efficient and be able to build and able to grow better crops and have better water. This can affect everyone incredibly greatly, mostly just because, we all want to live healthier. That should be, hopefully, the goal, is just to live good and healthy lives and then keep things better than when we found it. This is incredibly helpful for, the next generation, us, your children, because we also want the luxury of having something good, like good soil and good water in order to be able to drink. And we shouldn't have to be able to filter it out constantly multiple times. These are some of our plates from the soil section. We have some pretty nasty looking gunk. It all did very smell bad, I can confirm. But we were able to count them by looking at the different parts and sections. So on the PDA one, you have some really bright white dots, and you have some kind of more like gray, almost mucus looking stuff. And then you have some of the smaller dips that's just like a layer of kind of, a more see-through liquid, and that's also more fungus. And that's how we were able to classify and diverse the different types of fungus in order to put them onto the chart. Here are some fun pictures of us. The 3 pictures are where we took our samples at a wonderful, wonderful farm. We were part of the agricultural section, so we dug up some of the soil in between the rows to see what the microdiversity would look like. We also have some other pictures of that right there is the dam at the hatchery. That is where we took our water source from. And also there is me and Maya testing some of our water. The way that we tested it is we would put the water into this like kind of like a little smaller thing, a test tube, and then we would put different types of solutions into it, and we'd have to wait about either 5 or 2 minutes depending on where we were. And then you finally put in this like 2 drops of this like kind of smaller solution, and you'd shake it up and it would turn like bright blue. It was incredibly cool, and it would happen like super fast. That is the end of our presentation. Thank you very much for coming.